Hey guys, today I'll be talking about Treacher syndrome and what factors contribute to the disease. Firstly, Treacher syndrome is an autosomal dominant disease in which it results in severe craniofacial defects. The main cause of this disease is due to a mutation in the TOSF1 gene, and it has the genetic information that codes for the nucleolar phosphoprotein triacol. Triacol is important in the proper formation and proliferation of neural crest cells that develop the face. You do not know what crest cells are? Well, do not worry, we will get to that part later. Normally, when a protein is produced, the first steps occur in the nucleus where DNA is stored. The DNA double helix unwinds and RNA polymerase reads the template strand and adds nucleotides to the 3' end of the growing chain. This new transcript is called mRNA, and it leaves the nucleus and enters the cytoplasm to meet with a ribosome, where it would proceed to read the mRNA and translate it into a protein. Important note, ribosomes are made up of ribosomal RNA and other proteins. Now back to the TOSF1 gene. Remember, it encodes for the nucleolar phosphoprotein triacol, and it helps with the proper formation and proliferation of neural crest cells. Triacol also assists to facilitate the production of rRNA. However, since the TCOF1 gene is mutated, it results in the decreased function of triacol phosphoproteins. Since these phosphoproteins have decreased functions, it results in reduced protein synthesis to properly develop facial bones and tissues. To be much more technical, the insufficient production of ribosomes, also known as ribosome biogenesis, leads to increased neuroepithelial apoptosis, and the deficient formation of migrating neural crest cells results in the hypoplasia of the cranial skeleton, which can be seen in Treacher syndrome patients. But what are neural crest cells? Well, my friends, we are switching gears to embryology. Embryology is a branch of biology and medicine that is concerned with the study of, you guessed it, embryos and their development. We are going to focus on a trilaminar embryo as it represents an early stage in development of trioblastic organisms like us humans. And these two structures, the amniotic cavity and the secondary yolk sac, are the 3 germ layers: ectoderm, mesoderm, and the endoderm. During gastrulation, which is when an embryo transforms from a one-dimensional layer of epithelial cells into a multi-layered and multidimensional structure, the ectoderm is divided into the neural ectoderm and the non-neural ectoderm. This is important to know because after gastrulation, neural plate borders appear between the two. In the mesoderm, there is what is called the notochord. This structure is important as its secretions will go to the ectoderm, where it will accumulate to form the neuroectoderm and thicken to form the neural plate. Neural crest cells are then specified to the neural plate borders. They come closer together, which is when convergence is seen. The neural plate borders finally come together to form a neural tube which has separated from the ectoderm. The neural crest cells are then released and they migrate throughout the body and give rise to various cell types. They are important in the formation of multiple cells such as the chondroblast, neurons, melanocytes, smooth muscles, and osteoblasts. Now we are shifting to cell communication as there are signaling pathways involved in the formation of the neural crest, specifically the WNT signaling pathway. The early development of the neural crest can be explained by 2 steps. WNT signaling from the ectoderm and the mesoderm induce the neural plate border. And the second phase, WNT signaling from the ectoderm and the mesoderm, promote a set of transcription factors called the neural crest specifiers, and they help with the migration of the neural crest cells. The WNT pathway is important because it regulates cellular functions during development and adulthood. WNT proteins are lipid-modified glycoproteins which act as ligands with the frizzled receptors on the cell membrane, which help activate intercellular signaling. Quite similarly with the G protein-coupled receptors, frizzled receptors are 7-pass transmembrane proteins. There is also a co-receptor called LPR6, and when WNT is about to bind, it's going to bind with both receptors. There is a complex in the cytoplasm called the beta-catenin destruction complex. The proteins that make up this complex include DVL, axin, APC, and 2 enzymes called GSK3 and CK1. As you can infer from the name, this complex is going to destroy beta-catenin. In order to have the WNT target genes expressed, beta-catenin and transcription factor must enter the nucleus and along with TFC to transcribe WNT target genes. Normally, these genes would help with growth, proliferation, and development. But when beta-catenin is destroyed, that would not happen. This happens when beta-catenin binds with the destruction complex, and GSK3 and CK1 will phosphorylate the beta-catenin. This results in a series of steps in which beta-catenin will be destroyed via a proteasome. When WNT does bind to the LPR6 and frizzled receptors, the beta-catenin destruction complex is inactivated, and this is primarily due to the LPR6 because the two enzymes, GSK3 and CK1, interact with LPR6, making them unable to phosphorylate beta-catenin. This means that beta-catenin can enter the nucleus and help with the transcription of WNT target genes. Remember, this video is about Treacher-Collins syndrome, and as I said before, it is primarily caused by a mutation in the TOSF1 gene, which results in the decreased function of the nucleolar phosphoprotein triacol and protein synthesis of the ribosomes. What does this mean? So even if WNT is present, the mutation in the TOSF1 gene will result in a very low amount of functional ribosomes, meaning that the amount of WNT target gene proteins will be reduced. This results in increased apoptosis in the neuroepithelium, which also results in reduced migration of the neural crest cells, cells that play a pivotal role in the formation of the face and other tissues in the body.